Morgan gave rise in his lab at Columbia to a whole school of geneticists that laid down all of the fundamental rules of heredity that shaped the rest of the century, including all the fundamental rules we apply today in human genetics.
But what the genetic material was, how it was organized in the chromosome, precisely how the chromosomes replicated and how you replicated the genetic material, and what it actually does in the developmental process, that was a black box.
Scientists knew that what was in the black box could in some way be called the secret of life. The beauty and wonder of life itself. The incredible diversity that had puzzled and intrigued Darwin and Morgan. Could this be reduced to chemistry? Understanding the chemical basis of life had become the ultimate challenge for many scientists.
The fascination was, can you put together the ordinary atoms that chemists know, carbon, phosphorus, hydrogen, and others, and make something that's alive? What is the physical basis by which something can reproduce itself? How does chemistry and physics explain that? Maybe there's something new, a new principle that we don't know. Is it like that Michelangelo is a spark that has to go from one finger to another? Or what is it that makes something alive?
To be alive and grow, the cells in an organism must be able to reproduce and pass along their genetic information to the next generation. Whether it's a human embryo or a blade of grass. Morgan showed that genes transmit information from parents to offspring. But how? By the 1940s, scientists know that genes contain DNA, short for deoxyribonucleic acid. Either proteins, which do most of the work of the cell, or DNA must contain the secret of heredity. But which one?
All smart, right-thinking people at that time knew that DNA was an utterly boring structural molecule that couldn't possibly contain anything as interesting as heredity. Proteins had to be the stuff that contained heredity because proteins were very variable and very interesting. And yet the experiments kept pointing to the fact that it was DNA which, when transferred to a new organism, contained the secret of heredity.
In 1951, at King's College in London, researcher Rosalind Franklin takes X-ray photographs of DNA molecules. At 33, her scientific accomplishment is testament to her determination. She defied her father, a London banker who thought education was unhealthy for women and had wanted her to be a social worker. Instead, Franklin has become an expert X-ray crystallographer, and her latest photograph of DNA suggests a helical or spiral shape. Cautious by nature, she decides she needs more evidence before sharing her findings. She tucks the photo into a drawer. "60 miles away at Cambridge University, James Watson, a brash 24-year-old American geneticist, is also consumed by the study of DNA. As a boy, he starred on The Quiz Kids, a Chicago radio show, and by age 22, he'd earned his PhD. Working with Watson is British physicist Francis Crick, of whom Watson will write, "I have never seen Francis Crick in a modest mood. He talked louder and faster than anyone else." Crick had studied physics at University College London. but at 35 has yet to complete his doctorate. As they recall 20 years later, somehow it's a match made in heaven.
He thought like I did. Neither of us had sort of preconceptions. Neither of us were trained for what really interested us now.
As you said, we had the same point of view. And of course, I found the same reaction to you as you found to me. I was self-taught. I was stuck in Cambridge. There weren't that number of people who I could communicate with.
Later that year, on a trip to London, Watson is shown the X-ray Franklin is not ready to share publicly. My heart began to race, he says later. He rushes back to Cambridge to tell Crick that the shape of the DNA molecule may well be a helix.
They then began the task of literally constructing a model. Back in Cambridge, at the Cavendish, they took pieces and they tried putting them together in different ways.
Certainly they were working at a fever pitch.
Was it a race? Of course it was a race. Who wants to discover one of the secrets of the universe second? And they went after it with full force.
They come up with a model that looks promising.
I was very excited, probably because Francis was excited. It was our first DNA model.
It did look like a sort of cylinder of things, didn't it, Jim?
Sort of a solid cylinder.
With things sticking out, roughly speaking.
It wasn't very pretty.
We were completely on the wrong track. So when you have an idea and you've been enthusiastic about it, and it's clear it's wrong, you must just push it away and get rid of it.
DNA has 4 chemical components, or bases, called adenine, thymine, cytosine, and guanine, or A, T, C, and G. The bases seem to occur in pairs. So Watson decides to try something else.
I cut some things out of cardboard and sort of made the right shapes and then pasted things on which would indicate hydrogen atoms. Then I think I went off and played tennis. And it wasn't until the following morning I came in and I started to put them together in pairs.
When Watson makes models of the base molecules, he realizes that when they're combined, A with T and G with C, each pair has the same overall shape.
It all fell together. And when it fell together, it was so obviously right that they had to have known, they did know, that they had cracked one of the major secrets of the universe.
Watson and Crick suddenly realized that the structure of DNA is a double helix, with the AT and GC bases on the inside of the molecule, shown here as red and green, yellow and blue. The base pairs connect to each other between 2 ladder-like chains of sugars on the outside, all of it twisted like a double corkscrew. Watson and Crick published their findings in Nature in May 1953, sending shockwaves throughout the scientific world.
I was in the Department of Chemistry at Caltech, and Max Delbruck was in the Department of Biology. He asked, "What do you think of the paper by Watson and Crick?" I said, "I'd never heard of these papers." So he reached behind to a stack of reprints he had on a shelf behind his desk, and he threw the whole heap of reprints, because Jim Watson had sent him a bunch of reprints of the 2 papers, at me. And this floor is covered with these little reprints. And he said, "Read it carefully. The most important discovery in biology in 10 years. Later, I wondered why he didn't make it 50 years. But in 10 years, and you don't know about it.
In 1962, the Nobel Prize goes to Watson and Crick for discovering the structure of the DNA molecule. But Rosalind Franklin does not share it. She had died of cancer several years earlier at the age of 37.